Hi, everyone, and welcome to MacTalks. This is the first in a series of conversations with McMaster University researchers who are making a difference in the global fight against COVID-19. We are so delighted tonight to have our first MacTalks moderated by the Faculty of Sciences' first ever journalist in residence. Gretchen Reynolds is among the world's most read columnists at the New York Times. She's also a best-selling author who shares our passion for science and research. Tonight, Gretchen's going to be talking to Karen Mossman. Karen is McMaster's acting vice president research and a molecular virologist, as well as a professor of pathology and molecular medicine. Karen is among the outstanding team of McMaster researchers who've joined the fight against COVID-19. So now let's join Karen and Gretchen and go behind the lab door.
Hello, Dr. Mossman. It's very nice to meet you. I'm Gretchen Reynolds, and I actually want to start by speaking for all of the general public by saying thank you so much for all that you and your colleagues are doing and have done. We are tremendously grateful. And now I want to go behind the lab door. And I actually want to start by asking you what the lab was like before the coronavirus started. What were you working on, and how did that sort of play into your ability and your colleagues' ability to pitch into the coronavirus research?
Well, thanks for that, Gretchen. So my lab works on viruses. We have for the last 20 years, and we're interested in viruses from a number of different perspectives. We're interested in how viruses interact with the immune system. We're interested in how we can use that knowledge for novel cancer therapies in something called oncolytic viruses. And more recently, we got very interested in how viruses are different in different hosts, and particularly not just in humans, but in bats. Because bats, we know, can be infected with a whole range of different viruses and they don't get sick. So that's become, in the last probably year and a half, a huge interest in the lab, and it's that research that really prepared us the best for this current outbreak.
So you were sort of prepared for all of this, and then came the coronavirus. Had you and your colleagues studied coronaviruses? And then how did you get sort of looped into this particular area of research?
So we actually were gearing up to do some work with coronaviruses, and in particular, the MERS coronavirus. It's a great model for looking at a pandemic coronavirus. And
that's the Middle East.
That's the Middle Eastern respiratory syndrome virus.
And so when this novel virus first appeared in China, did you and your team start sort of paying attention then? Or what was the timeline, and what was your contribution? What were you doing in this particular area of research?
So we were keeping a very close eye out, especially when it was very soon after they recognized that the infections they were seeing in China were due to a coronavirus. It was very quickly understood it was likely a very similar virus to the SARS outbreak in 2003. Mm-hmm. And they also very quickly knew that the virus, the sequence, was very, very similar to bat coronaviruses, or similar viruses found in bats. And so that obviously very quickly piqued our interest. So we were following the literature very closely during that time. And then when we started to see outbreaks in patients that were positive in Canada, that's when we really got involved. There's a postdoctoral fellow in my lab who spent his PhD working on the MERS coronavirus. And so he has a lot of expertise in working with pandemic coronaviruses and pathogenic coronaviruses. So he was able to lend support to colleagues in Toronto that had access to patient samples to actually culture the virus and to isolate the virus from those patient samples.
Wait, and what does it mean to isolate the virus, and why do you need to do that, and how difficult is it?
So to do screening just to see if someone is positive, you don't need a whole intact live virus. You just need a sample that even has just a bit or a piece of the virus. And then we use methods to amplify or to grow or to make many, many copies. Whereas if you really want to study the virus, if you want to understand how does it grow, how does it spread, how does it cause disease, you need to have the actual live virus. And so that's much more challenging to get, especially out of just like a nasal swab or a patient sample. And you have to do that in what we call a containment level 3 lab, so that the safety of the public is ensured, the safety of the worker is ensured, and everything that you do under those conditions takes significantly longer, just because everything is done in a very, very precise and safe manner.
So you start by actually getting basically a sample of the virus from patients, right? Or at least you know people have been infected. And then that has to come to the lab, and you actually have to isolate this virus from anything else that they might have been infected with, so on and so forth, right? And then actually become able to grow it in the lab. How long would that normally take if we weren't in the middle of a pandemic? And how long did it take this time?
So obviously our clinical colleagues were very good at sampling, because with the 2 samples that Aaron J. was given, very quickly, I would say within about 2 or 3 weeks, he was able to isolate the virus and grow the virus up. So we had some tricks from other work that we and others had done in the lab about what sort of cells would most likely be good cells to use for this. And so we had some good sort of backhand knowledge about how we should really try to do this. And then, of course, with Aaron J. working specifically with coronaviruses, he knew all of the tricks on how to work specifically with coronaviruses. And so it actually didn't take as long as we thought it might take. As you mentioned, how do we know it's the SARS-CoV-2 virus? How do we know it's not another virus that patient potentially was infected with? There's so many different viruses that can cause similar symptoms. So that's, again, where we worked with some phenomenal colleagues we have, both at McMaster and in Toronto, to actually do the sequencing. And so then once we saw the sequence of the virus, we knew that definitely we had the SARS-CoV-2 virus.
And so what does this mean now for how you can use the isolated virus to test the testing, to test vaccines? What is it used for, and how important is this in the fight against the coronavirus?
So the very first thing that Aaron J. did was to grow up a large amount of the virus so that we could share it with colleagues across the country.
And this is in a dish, right? In a petri dish, is that?
It is very much like in a petri dish. So there's a layer of cells because viruses have to grow within cells. So yes, it was like in a petri dish. We wanted to make sure that the first thing we did was have enough virus that we could share with our colleagues, because we have some phenomenal colleagues across the country, not just at McMaster, but across the country. Only those, of course, that have the proper containment level 3 laboratories, because you have to work under those conditions.
And can you just really briefly explain what a level 3 laboratory means? Just how secure is that, and how is it kept secure?
So it's kept secure in a number of ways, not just, physically secure, but it also has the way the airflow and the whole design of the facility is designed specifically so that any pathogen at all, whether it's a bacteria or a virus, will be contained within the facility. So we work within specific, what we call biological cabinets or hoods. The airflow within the facility is designed in such a way that everything flows into the facility, will not flow out of the facility. There's many processes and procedures and different rooms you go through, and it's all designed to ensure that the person working with the virus is kept safe, but that in the event that there is a spill, that it's contained within the facility and does not get out.
So now that you can grow the virus in these, however, in the dishes in effect, can you use that then to do things like test the efficacy of potential vaccines, drugs, tests for the virus? What do you use it for now?
So there are a number of different things that we're going to use the virus for. My lab in particular, we're really interested in the basic biology of the virus. So we're really interested in understanding how it grows, how fast it grows, what cell types does it grow in? Is it only in lung cells? Can it grow in other cell types in the body? Can it grow in and kill immune cells? There's so many different aspects of the virus that we just really don't understand. And it's important to understand that, to really understand the clinical symptoms that we're seeing. Why are we seeing some of the clinical symptoms that we're seeing? Well, you need to understand the biology of the virus for that to make sense. So we're very interested in that. But then you also need the live virus so that you can test novel therapies, novel antiviral therapies, because you want to ask the question, does this new therapy stop the virus from replicating? And so you need a live virus to be able to do that.
You had said earlier that one of the things that you were interested in before the coronavirus was looking at things like how viruses or other pathogens might affect the immune system. So are you looking, that seems like a really interesting and disturbing area in terms of how the coronavirus seems to be affecting people. So are you looking into that area?
Those are the experiments that we're actually doing right now, or the royal we, Aaron J. and the students, and I live very vicariously through the people in my lab who are the experts. And those are the exact experiments that we're doing right now. We're trying to understand from both sides. How do human cells recognize and respond when they first see the virus? But then how does the virus counter respond? And that to me is the fascinating part of studying viruses. It's not just the initial response, it's the counter response and the counter response and the counter response. And that's where it becomes really interesting to study a virus in the context of the host or the mammal that it evolved in, because then both the host and the virus will have evolved a measure, a countermeasure, a countermeasure. But in a brand new host, that doesn't occur. So that's where it becomes really interesting to look at both the original host, but also a brand new host, and to start to think about how both have evolved over time.
So have you been looking backwards at the bats and whether this particular coronavirus originated in bats, and if that matters? Now that it's jumped to people, does it matter?
Well, it matters in that it's really important to understand both from the immune side and how different hosts will respond. But it is important to have a good understanding of where do these viruses come from. Because if you can understand where do these viruses come from, how do they go from one species into another species, that in some ways will potentially help protect for, maybe the next pandemic. So, a great example of this was several years ago with one of the influenza virus pandemics when they realized that having, different species like birds and, say, pigs in very close proximity was allowing the virus to jump between those species. And so
Right.
then you can start thinking about, well, how do we ensure in some locations that we don't have those species very, very close to each other? So the more we can understand about how these viruses can jump from different species, the better we can understand where did these pandemics come from, but also are there ways that we can try to prevent that from happening in the future?
And so, in terms of where we are now, how much do you think we do know about this particular coronavirus, and how mysterious is it still? And how close are we to having something like an actual vaccine?
So the virus, to me, as a virologist, is actually quite interesting because as we learn one thing, we have two new questions. So for everything that we're starting to see and understand, it opens up more and more questions. So in some ways, it is very similar in sequence, in structure. If you were to look under an electron microscope, it would look almost identical to the original SARS coronavirus from 2003. But yet even from the pandemic, it's very different. The original SARS had a much higher mortality rate, but it didn't spread nearly as far or as quickly. So we didn't get near the number of people infected, but the overall mortality rate was slightly higher. So it becomes really interesting to see how is it similar, how is it different. And now that we're moving beyond the initial infection and we're getting now to see what happens in the protection. Do we see herd immunity? Do we not see herd immunity? Are people that were infected, are they protected? Are they getting infected for a second time? And so as the pandemic progresses, we start to see different clinical signs, we see different outcomes, and that, of course, starts to open up a number of new questions that we'll have to start to work on.
And the virus itself, is there evidence that it's changing, that it's mutating the way flu viruses often do? And is that a concern?
So due to the particular structure of this virus, it won't change and mutate in the same fashion that influenza does. Just because
That's
of the mere
encouraging.
structure of influenza. That's a unique sort of structure. So we won't see the same sort of genetic shift and genetic drift that we see with influenza. But now that more and more sequence data and more genomes of viruses, not even just within one geographical location, but over time, but also looking at different geographical locations around the world, within a certain region, we're starting to accumulate a lot of data on sequences of these viruses. And so we are starting to see that it is changing. It's not changing nearly as rapidly as some viruses can, which is good, but we're still seeing changes. So the viruses that we isolated here are still different than the ones from China, than the ones from Australia, but they're pretty similar.
And so what does that mean for something like a vaccine against this? Even more than drugs, it seems like the vaccine is the thing that will really change the arc of the pandemic. Where do you see us in that search, and where does your research play into that?
So there are so many groups around the world working on vaccines. So I'm the optimist, so I remain optimistic. But the reality is we don't yet have a vaccine against any coronavirus. Right. So we do not have a vaccine against the original SARS. We don't yet have a good vaccine against the MERS coronavirus. And sometimes there are viruses that it's fairly straightforward to make a vaccine that has high efficacy and provides really great immunity. And then, of course, there's some viruses we don't have a vaccine for. HIV, we don't have a good vaccine. Hepatitis C, we don't have a good vaccine. And so it really comes down to each particular virus and the biology of the virus. And so that's where really understanding the biology of the virus becomes exceedingly important.
And I actually want to get to questions from our listeners in just a moment, but I have one more for you, which is there something really strange about this virus in, for instance, the really wide range of host responses? I mean, some people virtually have no symptoms. Some people become extremely ill. Is that unusual for a coronavirus, or is that fairly common and we just don't understand that?
Well, in some ways, it's common with many viral infections that if you have a strong immune system, you'll respond much better. So in general, what we see is it's the elderly, the very, very young, the elderly, people that have underlying conditions that tend to respond the poorest. And not just to this coronavirus, but to a number of different infections, because their immune system is somehow compromised. And we're certainly seeing that, the elderly population is a very vulnerable population. But that doesn't explain everything that we're seeing. We are seeing some individuals that are, young, healthy, have no underlying immune deficiencies that we know of, and yet some of them are getting very, very sick and requiring hospitalization and ventilation. And so that's what we don't understand. And we don't understand, were they just exposed to a very, very high amount of virus, whereas other people are not? Or is there some, again, underlying way that the virus, specifically this virus, interacts with the host that we don't yet know?
And I do know there's some issues with people seeming to have a huge overreaction of their immune system, which again seems very variable. Is that unique to this virus, or is that also fairly common? We just don't hear about it a lot.
Well, that was certainly something that was seen with some of the influenza pandemics, where it wasn't so much the virus that killed some individuals. It was what we called a cytokine storm and an overreaction of the immune system that actually led to the side effects and the detrimental effects that actually caused individuals to succumb to the infection. So again, every virus is a little bit different, but there's certainly trends that we see with different viruses, especially pandemic viruses.
I've been receiving the questions from our listeners as we've been going on, and there are a whole bunch of them. So I will start with just the one that I think a lot of people have, which is, in your opinion, Dr. Mossman, when do you think we might get back to semi-normal activities, seeing family, friends, and gathering? We won't hold you to this timeline.
I would love to say within the next month or two.
Really?
I mean, in many cases, I think it will be regionally dependent. We have certainly seen some areas, either within our own country or other countries, where social distancing has been working remarkably effectively. And of course, we've seen other areas where that has not been as effective. And so I do believe that the best, and this has been played out ever since the virus first hit in China, and it certainly played out in all jurisdictions, that the best prevention is the physical distancing. So I do believe it will be, dependent on if you live in a very, very populous location, if you live in a location where, physical distancing is much easier to obtain. But I do personally believe that will be the driver, because that has proven to be the best prevention.
And a somewhat related question. Do you anticipate effective virus treatments to be available for the sickness before we get a working vaccine?
Oh, that is a great question. The reality is, I would guess that we probably will have antiviral drugs before we have an effective vaccine. That's
Why?
if I had to guess, that would be my guess. I mean, we already have data that actually just broke in the last day or two from a study in the States with remdesivir, which it still needs to go through the full review process, but the preliminary results from that clinical trial coming out is looking very promising. Now, that's a single drug, and certainly what we understand, again, with HIV and with others, where we have antiviral drug cocktails, a whole cocktail of drugs usually work much more effectively than one single drug. But the fact that we are seeing at least what appears to be some efficacy with some drugs like remdesivir, which even though it might not be the magic cure, I don't think we will have the one drug magic cure. But if we can find even a cocktail of drugs that each on their own are okay in combination, I predict, but this is my personal prediction, that will occur probably faster than what we will have a widespread vaccine.
But by and large, a vaccine is the aim, right? I mean, it's great
Absolutely.
to be able to treat people, but the goal is to prevent this. And right now that means social distancing, but hopefully in the future it does mean a vaccine, right?
Absolutely. The vaccine is the preventative. It's better to prevent than to have to try to treat. And we'll need both. The reality is we'll need both. Not everyone will take a vaccine. Not everyone is medically able to take a vaccine. So we'll definitely need to have both, but absolutely. Preventing a disease is much easier than treating a disease.
A couple of people have asked about the that has been involved in getting to this point in isolating and growing the virus. How important has collaboration been, and how important would you say this has been for furthering collaboration worldwide in science?
So collaboration is absolutely key. We would not be where we are if we had not collaborated. It was a combination of our clinical colleagues in Toronto that were seeing patients, that were able to, bring those samples to a laboratory at the University of Toronto. And then, Aaron J. from my lab having the knowledge and being able to go, they were very quick in allowing him the training and access to get into the level 3 facility in Toronto. We can't do anything without collaborating. No one lab can be the final stop. And we really need to collaborate with people that do the sequencing. And what I have really seen, which has been fascinating to me, and I hope it continues beyond this pandemic, is the willingness of scientists around the world to share, to show and put their data out in the public domain. Normally, you hear, publish or perish. You want to make sure you don't get scooped. So people are very leery of sharing any data until they have it published because they don't want to get scooped. They want that high publication. But what we're seeing now is people putting their data on preprint services. It's out almost as soon as it's generated. There are caveats. It's not peer reviewed, so you have to, evaluate it very, very closely. But it's that sense of it's more important for the community to have access to the data than it is for me to publish in a high impact journal and not get scooped. And it's been a wonderful for our field. Some fields do this. Our field has not historically done that.
Well, that brings up a question that I wanted to ask you, which is if you could give people advice out in the general public for how to evaluate some of the information that they're seeing, whether it's in a newspaper or on Twitter or from Facebook, what advice would you give them for evaluating the accuracy and reliability of the information that they're seeing about coronavirus? And I'd say you can rely on the New York Times. We try hard, but apart from that,
Well, it is challenging because even in some scientific journals, there are some scientific journals and some that do go through a peer review, and even once information and data have gone through a peer review, in some cases you only know what you know at the time, and so you make the best interpretation of the data that you have given your ability and your knowledge at the time to make that interpretation. And of course, over time, we've even found with a lot of our own data over time, as either techniques get better or just the general knowledge in the field increases, all of a sudden we have a different interpretation because we know more now than what we knew then. But the first thing I would say, especially nowadays, is if it sounds too good to be true, it probably is. If it sounds, a little bit crazy, it probably is. Early on there were, hold your breath, and you can hold your breath, then, you don't have the virus. I mean, you have to use your own judgment. And certainly when we're evaluating data, we also look at where is the source coming from. Mm-hmm. We certainly know, especially for viruses like this, who are the labs that are virology labs? Who has a history of working with these sorts of viruses? Who have the history or the collaborators that are well known and well respected? And so then that gives us a lot of comfort when we read, new data that has not been peer reviewed, if it does come from, again, a very reliable source. So same thing in journalism. There are some magazines that you rely on and you trust much more than others. And that's the same for us. There's certainly leaders in the field that we feel very confident that they've made the best interpretation that they can with the information that they have.
Do you get the feeling that this pandemic has helped to restore some of the public's basic trust in science? And do you think there had been some degradation of that? So certainly it felt that way in the United States. So
I'm not convinced it's restored everybody's faith in science in some areas of the world, but I think overall, I hope it has. I mean, I hope people are recognizing that, we are all of the scientists, all of the, even in engineers and social scientists, and it's not just people in the medical field, it's our economists, our public policy colleagues, our social scientists. We're all doing everything we can to try to figure out how to find a cure, how to make that cure affordable, how to make sure that cure can get to the most vulnerable populations, how to make sure the policies are in place so that it can easily get there. How do you ensure that the knowledge goes with that? It's a multidisciplinary problem, and everybody is working together to try to solve that problem as quickly as we can. And I hope the public sees that.
This person is asking a little bit of the opposite of that, which is, if we could rewind the clock by 2 or 3 months, what could have been done differently in terms of preparing for and responding to the pandemic in Canada?
Oh, wow, that's a great question. I mean, hindsight is 2020.
Yeah, it always is, yes.
In some ways, I think we did a good job. We had the advantage of seeing what was happening in China and then how that was spreading to places in Europe before it came here. So in some ways, I think we did respond faster than some other countries did because we had the luxury of seeing it sort of unfold. I don't think we were prepared for how quickly it could spread because, again, it's very different from our experience from SARS, the original SARS coronavirus. I mean, there was a huge outbreak in Toronto, but for example, it didn't hit Hamilton. Right. So again, you learn from every single pandemic you go through, but some of the lessons we learned from the original SARS don't apply here because the virus is very different. But I think what hopefully we'll learn for the next is, we should be prepared with adequate PPE. And it's not good enough to stockpile huge amounts, but let it sit there for, 10, 20 years so that, then the quality expires. I mean, so I think there are some things that hopefully we will learn now from this for the next one.
Wait, we only have time for a couple more questions, but several people asked one version or another of, why is it so hard to make a vaccine for this particular or any of the coronaviruses?
So there's a number of factors to consider when making a vaccine. First of all, what part of the virus is really going to generate a strong immune response? Right. And we won't know that until we actually start testing. So it's hard to predict up front how the immune response is going to react to any one bit of the virus. We can have some educated guesses based on similar viruses. We know what's on the outside of the virus, so we know what the immune system should be able to see. But it's until you actually do all of the testing, it's really challenging to know just by guessing is how will the immune response actually respond to different bits of the virus. And many viruses also have ways of turning immune system on or off. So we really, it's not as simple as we have the sequence, we can make a vaccine. There's so many other factors that play a role.
And several people have asked, so how has your lab sort of handled actually two parts to this. First of all, how is your lab and you personally, how has this affected your life? Can you still go home, see your family? And secondly, what has it been like to get the kind of attention that you have deservedly been getting recently?
So for me personally, I have been working in my home office for the last 6 or more weeks. I am not considered essential within the actual lab. As I said, I live vicariously through my students and postdocs who are the real heroes. For Erin J., who is really the star of the lab, who has that expertise working with pathogenic coronaviruses, he has been just absolutely amazing. He lived with one of our colleagues in Toronto for, and he's been newly married, but he lived in Toronto for probably a month or so that he could basically work around the clock in the facility in Toronto, going back and forth for a while between Toronto and McMaster. And now that we have our level 3 facility set up, he can now work at McMaster. So for the
you have one set up there too. That's great.
So we do have the facility and here it takes a little bit of time to make sure it's all set up and ready to go for this particular virus because of all of the safety areas. But now we have the virus at McMaster. We can work a number of colleagues safely. And so, for Erin J., that makes life a lot easier. But for those that it's like all of our healthcare workers and everyone on the front line, it's exhausting. They have to wear full PPE all day long when they're in the facility. It's very challenging going in and out of the facility. So once you're in for hours at a time. So for the ones really doing the hands-on work, it's like our front line workers in the hospital. It's challenging, but it energizes them and it's exciting. Because this is what, as Erin J. will say, he always wanted to be a virologist. And even as a kid, he said, I want to be the one to help solve a pandemic. So this is what he has spent all of his training for. So for him, it's the highlight of his career, and he's just phenomenal.
Well, this will be the last question, and builds on what you just said. Several people have asked a version of, so what's the next big research question? What do you see sort of on the horizon that you might already be preparing for?
Oh, there's so many different questions that, as I said, we answer one question and we come up with 10 more.
None of which might be the question that actually emerges, because who would have thought that the coronavirus would take over everything? So,
I mean, I'm still fascinated because my whole research career, I've been fascinated with how viruses interact with their hosts. And so I'm still fascinated by how we can have such a different response when the virus gets into a human cell versus into a bat cell. Bats are mammals. Their immune system is very, very similar to ours. The pathways are almost the same. The proteins are almost the same. We are starting to find really, really small, minute differences, but that can change the entire course of an infection. So for me, that's what's fascinating, because the more we understand how that works, the more we can start using that knowledge to generate the antiviral responses and the therapies to basically make our immune system look more like a bat immune system, because they control these viruses exceedingly well.
Well, on that note, although I have dozens of questions both on my phone and in my head that I'd love to ask, we are going to have to end. But again, all of us out here in the public are so grateful for all that you are doing. So thank you. Please keep up the great work and let us know as it all proceeds. Thanks very much. Thanks everyone for joining us.